All right, welcome everyone. Today we're going to be talking about comparative genomics. So first part of comparative genomics is using evolutionary signatures for genome annotation. And we're now entering the fifth and final foundations module of the course before we get to module 6, which is current research directions and guest lectures as well as sort of emerging topics. So today we're talking about comparative genomics. And then on Thursday, we're talking about comparative genomics for understanding genomes. So basically using evolutionary signatures for interpreting genomes. And then on Thursday, we're going to be talking about using comparative genomics to understand evolution itself. So we're going to be flipping it around on its head. So basically today, we're starting with comparative genomics and we're starting with evolution and we're using it to understand genomes. And then on Thursday, we're using genomes to understand the very mechanisms of genome evolution. So we're going to be talking about genome-wide alignments, we're going to be talking about structural changes, about genome duplication, and so on and so forth. And then next week, we're going to be getting much more into evolutionary theory, into phylogenetics, and sort of the relationship between many different species, into evolutionary rates, models of evolution, and how to take those dynamic programming approaches that we saw in linear fashion and the sort of hierarchical fashion of RNA structure, and then now using it on nested trees to basically infer phylogeny on in a Bayesian framework. And then next Thursday, we're going to be talking about phylogenomics. So basically, this is phylogenetics at the genome level, namely beyond just one gene at a time. How do we leverage the whole genome? And that's very similar to the way that we ended the genetics part with systems genetics. Basically, how do we leverage the whole genome to understand heritability and these global metrics? In the same way, phylogenomics is going to be finishing this part at the whole genome level. And then that's it. Then we have our quiz, our in-class quiz. I like to say that it's not a midterm. It's much cuter than a midterm. It's a quiz. And that's sort of where we're sort of stopping the formal presentations and then, going into single-cell genomics and then human phenotypes, cancer genomics, genome engineering, and sort of all of the frontiers of the field. So what's this module about? This module is about leveraging evolution to understand genomes and vice versa. I showed you this slide at the very beginning of lecture 1. I basically said, hey, we can actually use comparative genomics to recognize important parts inside genomes. And I showed you this snapshot from my own thesis, which basically showed the conservation of 4 yeast species and how this could be used to elucidate islands of conservation, which I've underlined here, and use these islands to then recognize regulatory motifs, to recognize functional elements within genomes. And all of the previously known regulatory motifs in this region are shown here in boxes. And you can see that there's a very good capture of these elements by these underlined regions of perfect conservation. Everybody remember this picture? Great. So the whole concept here is that we can actually read evolution to reveal functional elements. And that's what we're going to be talking about today, how we can use evolution to study genomes. And then on Thursday, we're going to be doing the converse, basically using genomes to understand evolution. Okay, and it's a feedback. The better we get at understanding genomes, the better we can understand how evolution is acting on these genomes. And the better we understand evolution, the better we can understand the genomes themselves. So we're going to start simple and sort of increase in complexity like we do in most lectures. So first, we're going to be talking about nucleotide level conservation and measuring evolutionary constraint. We're going to introduce the concept of purifying selection, neutral branch length, discovery power with more genomes, and how to detect constrained elements at the level of individual nucleotides, at the level of windows, and also building a hidden Markov model. And then we're going to also go beyond the detection of elements and see how we can estimate the fraction of the genome that is conserved and some basic principles of signal versus background separation, which are quite pervasive in machine learning beyond just this particular application. Then we're going to talk about evolutionary signatures, where we're going to be focusing not on the amount of conservation, but on the pattern of change So the concept will be that different types of functional elements in the genome evolve in very characteristic ways. And by reading these characteristic patterns of evolution, we can in fact recognize and distinguish not just quote unquote important regions, but different classes of regions. And we're going to distinguish protein coding genes, non-coding RNAs, regulatory motifs, transcription factor binding sites, microRNAs, through the use of different evolutionary signatures. Everybody with me so far? So, and then we're going to look at how we can actually measure selection within the human lineage, not just across species. And that's much less branch length, much less power. But when you aggregate many regions together, such as regulatory motifs, different classes of elements, then you can get sufficient power by leveraging the whole genome. Everybody excited? So comparative genomics has really come of age. There are dozens of projects that are sort of sequencing different parts of the, fauna and the flora of our planet. So in the mammalian world, the Drosophila world, the fungal world, there's been really so many different projects and so many different sequences and resources that are widely applicable. So if you guys are interested in comparative genomics, now's a fantastic time to go after all of these sequences and what can be read from them. And the concept of these projects is that you want to compare related species in order to discover conserved and important functional elements, okay? The concept is very simple. It's basically embracing Darwin's evolution. The concept of random mutation followed by brutal selection, okay? So mutation is completely blind. It just like puts mutations everywhere. And selection is completely brutal and it only keeps the good stuff, okay? And these are the two forces that are shaping evolution. So over evolutionary time, non-functional regions will be tolerating these mutations. Basically, when natural selection comes about and says, hey, who's the fittest? Who can survive? Any mutations that accumulated in functional regions and therefore disrupted these regions will lead to less fitness, lower fitness for the individuals carrying these mutations. So those individuals over many generations will basically have fewer children and these genes that carry these mutations will eventually be weeded out from the pool of genes. That's in functional regions. Basically, these accumulate mutations at the same rate as non-functional regions. Both accumulate mutations. But these functional regions decrease fitness. But in non-functional regions, mutations can be perfectly well kept. Okay? And then, over evolutionary time, the less fit organisms and therefore their genes get thinned out. So any mutations that accumulated by disrupting these functional parts of the genome will be weeded out. Raise your hand if you're like completely with me here. Awesome. Of course, there's also the concept of positive selection. Some of these changes will be advantageous. Very rarely, but they will be. So a tiny, tiny fraction of the mutations that happen in a genome are actually conferring an evolutionary advantage. And those mutations, again, over evolutionary time will rise in frequency, unless there's some massive selective event that kills off everybody who doesn't carry that mutation, and then they rise to fixation much more rapidly. Everybody with me so far? So these are the two forces. Basically, mutation is completely random, hits everything at once at the same rate, and then selection weeds out the bad stuff. So basically, when you see this picture, it's not that mutation is super careful not to disrupt these motifs. It's that mutations have happened everywhere, and simply those mutations that disrupted these regions were weeded out. And therefore, over evolutionary time, when we compare these species, we find that those regions that have excluded mutations are the ones where functional things were disrupted, and therefore these mutations were lost. Sounds good? It's amazing how many adults still get this wrong, so I'm beating you over the head with it. So that's sort of the first principle, that over evolutionary time, mutations tend to be kept preferentially in the non-functional regions and then tend to be excluded preferentially in functional regions. Now, the question is, what happens across larger and larger and larger distances? So basically, all these projects that I mentioned earlier are sequencing dozens and sometimes hundreds of species in each of these lineages. Why? The reason is we want more power. And the idea is that with more branch length, you have more opportunities to observe differential events and therefore more power. Basically, if your goal is to distinguish functional versus non-functional nucleotides based on the number of mutations that accumulate, with very close distances, you simply have no mutations in either region. You basically compare two regions between human and chimp, you're like, well, no mutations here, no mutations there. You know, there's not much you can do. Whereas with sufficient distance, the ability to distinguish increases. That's sort of when you start shedding light on the two regions. But the problem is that with very, very far distances, if you now start comparing human and yeast or human and amoeba and human and bacteria, the functional regions are no longer conserved, okay? So basically, there's increased power with increased distance, but at some point, these species are just simply billions of years apart. Their environments are dramatically different. They have adapted in different niches, and very few of the common regions are still preserved. And that's where the many closely related species strategy comes in, okay? And that's much, much better than simply few distantly related species. Yes, it costs more because you have to sequence more specimens, but in the end, it gives you much greater power. Basically, for the same total branch length, you prefer many close species because the functional regions will be much more likely to be conserved for each pair of species. Say 20 million years have elapsed or 60 million years between all mammals. That's, you know, relatively little time in the, you know, scale of evolution, where basically we've been evolving for more than 1 billion years on this planet. So 60 million years is really just a blink of an eye. And all mammals have basically preserved some basic common functions, like eating and breathing and, frankly, thinking and planning and seeing and hearing and digesting and all of that stuff. So basically, for nearly all functions that make us, you know, alive and well, other mammals have them too. And the corresponding functional elements will also be preserved, okay? So functional regions are conserved for every pair of species, and the non-functional regions, and that's what's really cool and important, accumulate the noise independently, okay? So in every one, if each of us were, I don't know, a different mammalian species and we all diverged 60 million years ago, every person would accumulate different mutations over these 60 million years. So by sequencing 200 mammals, we basically have 200 different opportunities to observe mutations in non-coding or in non-functional regions. Whereas the functional parts will be minutely preserved in nearly all of us. Everybody with me? So it's kind of like recording audio from many different places in the same concert hall. So you may have, I don't know, somebody chewing chips over here. It's like, I don't know, Peter and the Wolf. Basically, you go with your kids, you know, one kid is crying over here, some kid is like eating crisps over there, some, I don't know, older gentleman is telling his wife, this is great, over there. And then you miss different parts of the orchestra at each different part, but all of that noise is independent, whereas the signal is preserved in all of the spots. And therefore, by having multiple recordings, you have multiple opportunities to eliminate the noise. Everybody with me? So again, having multiple microphones can really help in the concert. And that's where, if you were to only look at, you know, 2 species, and you went out to an average of 4 substitutions per site, that basically would mean that almost nothing is conserved, that you're basically going to yeast distances. But what you're actually doing is accumulating more and more species. So if you have 3 mammalian species or 10 mammalian species or 20 mammalian species, you now can get very quickly to a huge number of bombardment events and therefore have much more power to distinguish functional and non-functional regions. Everybody with me? Raise your hands if you feel you've learned something already. Yeah? Awesome. Great. All right, so now that we've sort of understood these basic concepts of how selection actually works, how this branch length increases with evolutionary distance, and how discovery power increases as well, let's get down to the nitty-gritty detail. Again, we're going to be talking not about overall, basically when we measure this overall branch length, this is the neutral branch length. Of course, in protein coding regions, you're not going to have 4 substitutions per site. But in non-functional regions and in most non-coding regions, you're going to have about 4 substitutions per site. So basically, this is the neutral branch length, because we don't measure branch length in the functional regions because these are under constraint. Okay, so now let's use that to detect constrained elements. First, talk about nucleotides and windows and an HMM approach. So as I mentioned earlier, because we're comparing closely related species, few events have happened. And that's not just at the nucleotide level, that's also at the global rearrangements level. So if you look at these species of Drosophila, which are roughly at the same distance as different mammals, you basically see that the different genes are more or less in the same order. So that basically means that I can construct genome-wide alignments, not just for the protein coding regions, but also for the non-coding regions. And that's extremely important if you want to discover not just protein coding genes, but also non-coding elements. And this collinearity and conserved order spans over hundreds of genes. And sometimes you will have a breakpoint and then we'll just reshuffle these segments and sort of put them back in the same order. And that allows us to basically build these genome-wide alignments and use comparative genomics to identify functional elements. And within these regions, we end up with a picture like this, which I also showed you on the very first lecture. The concept is that you can pile up more species like dog, mouse, rat, chicken, fugu, zebrafish, and then align them all to a same common reference. In this particular example, it's human because we are so self-centered as scientists. And then in blue, you see the annotation of protein coding exons in human. They're the tall blue bars. And then the thinner, the less tall blue bars are the UTRs, the untranslated regions. 3' UTR and the 5' UTR, okay? What do you notice in this picture? What you basically see is that as you start looking at the different species, some elements are much more deeply conserved than others, okay? So tell me some observations that you're noticing. What's one observation? Almost all the exons are very deeply conserved. What's another observation? Yeah. The introns are more conserved, more closely related. Exactly. So, moreover, while the exons are very deeply conserved across all vertebrates, the introns also appear to have conserved elements, but only within the mammals. As soon as you go outside mammals, chicken, fugu, and zebrafish don't have the same conservation in introns. What's another observation? What's going on between exons 5 and 6? Yeah? Another gene is not in, or another exon is not in. There appears to be something else that's very deeply conserved, just like very strongly conserved, just like an exon. How deeply conserved is it? Only in the mammals. So, is it a new protein coding exon that arose in the mammals? Or is it a non-coding region that happens to be super strongly conserved, just like the protein coding exons, right? So how do we find out? We can go and knock it down. We can basically, see whether it makes a transcript, whether it's protein translated, whether it folds as a non-coding RNA, and do all kinds of experiments. We could basically pay, I don't know, our buddy who's running a company that does experiments for service, some fee for each type of experiment and just go do them all, right? What else could we do? Well, we kind of have already paid evolution, right? Evolution has done gazillions of these experiments. We could just ask evolution, hey, can I look at the notebook, your lab notebook of sort of what happened with each of your experiments? And that's what we're going to do today. We're basically going to, instead of looking at the experiments that our buddy can do, and then sort of, you could test it in, but maybe that exon is a protein coding exon, And it's only expressed in the outer layer of your retina, right? So what's our body going to do? He's going to grind up a bunch of mice and then see where do I see transcription. And he's not going to find it anywhere because the retina is like 10, 20 cells out of your, whole mouse. So, he's going to look for proteins, but maybe it's only expressed, during early development, and then it stops, and so on and so forth. Okay. So basically, experimentally, it's very hard to find out what every single region of the genome does. But evolutionarily, even if this is only expressed for 3 days during the developing mouse, if you lose that element, the mouse can no longer see. Do you think this mouse is going to have a lot of offspring? No. So that's what evolution is helpful for, okay? So basically, we're finding that exons are deeply conserved to mouse, chicken, fish. Many other elements are also conserved. Are these exons, are they regulatory? So, as we can see, there's all of these big islands of conservation, and some of them are in fact, protein coding or known to be protein coding, and others, we don't know whether they're protein coding or not. So we're gonna find out. So basically, what we're gonna do is estimate, number one, the level of constraint, but two, estimate the type of constraint. So how do we go about finding the level of constraint? As we talked about in the early lectures, you could do many different things. You could, number one, count the number of edit operations, the number of substitutions and gaps. You could also estimate the number of mutations, including back mutations, as we're going to see on Tuesday. You can incorporate information about the neighboring neighborhood. You could basically look for conservation windows. You could estimate the probability of a hidden conserved state or a hidden non-conserved state, and then alternate probabilities. And then this is what we saw, the week after we first introduced this slide. You could use phylogeny to estimate the tree mutation rate or the rejected substitutions. And this is sort of one of those techniques that we're going to see in phylogenetics. And you could also allow different portions of the tree to basically have different mutation rates, okay? So you could basically say, well, this region has a faster rate, this region has a slower rate, and so on and so forth. And, phylogenetic techniques are going to allow us to do all of that. So to get at these conserved regions, you could look at two different things. You could look at the overall rate of change, okay? So here's one example of a neutrally evolving sequence, and you can see that at every column of the alignment, if I have the same sequence as the reference, in this particular case, the reference is mouse, then I put a dot. So basically, this is all A's, all C's, and so on and so forth. And then as soon as I have a mutation in one of the species, I just draw out the mutation. I just write out the mutation. So what do you notice here? You notice that in a neutrally evolving sequence, all of the nucleotides are equally likely to be replaced by any other nucleotide, okay? Here's an example of a sequence that has a decreased rate of change. So you can actually build a model of your substitution rate. You could estimate the maximum likelihood estimate of your substitution rate, or the maximum posteriori estimate, by having some prior distribution for what evolutionary rates look like across the genome. And then you could report both the rate that you estimated, that maximum likelihood estimate, and also the log odds score that rate is non-neutral based on the total number of substitutions. If you only had one column of the alignment, you could say, well, I only have so much evidence. If you have many, many more columns of the alignment, you could have a more robust estimate and a higher probability, or a higher p-value, a higher log odds score that your sequence is evolving in a non-random way. And you could do that both at every nucleotide in isolation, or you could do that in a window-based approach. Raise your hands if you're with me so far. Awesome. So that's the rate of change. Everybody with me? So the other approach is to look for the pattern of change. So if I look at this sequence here and I measure the rate of evolution, I will find that this has not changed at all, this hasn't changed at all, this has a 0.8 rate, this has a 0.5, 0.6, 0, 0, and this column here has 3 substitutions, a much faster rate, okay? So it appears at first glance that this nucleotide position is non-functional, completely free to change. But what do you notice that's funny about it? Every single time it has changed, it has changed always to the same letter. Okay, this is kind of weird. Moreover, this is the phylogeny of the species, and these substitutions are not all clumped in the same subtree. So you can't just say, oh, there was a C to G change, and then all of these guys maintain the C. Okay. Instead, there's alternations between C and G, C and G, C and G, C and G. So that basically means that there were multiple independent events switching that C into a G. What is the chance that this would happen randomly? Very small. At each position, assuming that there was a change, you have 1/4 chance of changing into or 1/3 chance of changing to a G. You assume there was a change. And then times 1/3, times 1/3, times 1/3, times 1/3, okay? So it's very, very small probability that this is just by chance. So what is probably happening is that there's constraint at that position to preserve either a C or a G. And this also tells you that there's mutations happening everywhere, and these are simply excluded by natural selection. All of these mutations that change these functional positions were excluded, but here, all of these different mutations that happened were, evolution didn't care about. So it kind of kept them. It didn't find any flaw in the resulting functionality of that region. Is everybody with me here? So what we can actually distinguish is unusual substitution pattern, not just rate, okay? So we can build a probabilistic model of some stationary distribution that is different from the background. And you can basically build a maximum likelihood estimator, which is a vector of nucleotides that are preferentially conserved. And this is the representation of that vector as a position weight matrix. Okay, so you can then report the log score that that vector is non-neutral. And here you can see that this A is very strongly conserved, and then this C or G is, perfectly fitting. And then you will notice that, in fact, this is a motif for a transcription factor. Okay, so in fact, what is happening here is that evolution is minutely preserving that pattern that matches your motif. Everybody with me here? So you can actually do that with more species. You can reveal individual transcription factor binding site. As you start piling up hundreds of mammals, you can basically identify individual motif instances, and then you can sort of corroborate those using both evolutionary constraint, but also using matches to known motifs, and also using transcription factor binding using chromatin precipitation that we saw in the motif lecture. So basically, chromatin precipitation for the NRSF binding factor will basically tell you that there's all of these instances of binding of NRSF. Indeed, the motif match is there, and indeed, all those positions are preferentially conserving the NRSF motif. Who thinks this is all kind of cool? Awesome, great. Okay, and then the reason why this is really important is you can then go into GWAS that we talked about last week, and you can basically ask, are these SNPs that are associated with disease preferentially disrupting conserved elements? And in this particular case, this SNP here, this A2G mutation at that location, which happens to be in an enhancer in K562, an immune enhancer, is in fact disrupting a motif for FOXO, which is one of the regulators active in that cell type. And you can basically see that because of all of these excluded substitutions across different species, okay? And therefore you can say, well, great, there was a mutation here, and that mutation is particularly disruptive to that motif. So functionally associated SNPs are enriched for particular chromatin states and for constraints, and that allows you to prioritize candidates, increase the resolution, and also recognize disruptive motifs. So that's all trying to recognize individual nucleotides and individual windows that are under constraint. But what if you wanted to say what fraction of the human genome is under constraint? Then you could, of course, ask what fraction can I detect to be under constraint? But that number is smaller than the fraction you can actually estimate to be under constraint. And the reason is the following. Every single time you can detect a portion of the genome as being under constraint, you choose some threshold. And everything above that threshold you call constrained, and everything below that threshold you call non-constrained. And with 4 mammals, you can basically set that threshold at some level with some false positive rate, say 5% false positive rate, false discovery rate. And then you can detect 0.6% of the genome as indeed conserved. With 29 mammals, you can detect a much bigger fraction of the genome as being conserved. With 200 mammals, we can actually recognize the conserved positions for an even larger fraction of the genome. But then if you estimate what fraction of the genome appears to be constrained, and you can do that at the level of 50 nucleotides or at the level of 12 nucleotides. At the level of 50 nucleotides, you don't recognize much. And part of the reason for that is that it might not be in 50 nucleotide chunks, okay? And part of the reason is also that the smaller your element, the less power you have, because you have fewer opportunities to observe events. And so as you increase the resolution to a 12-mer, you completely lose your signal in only 4 mammals. But with 29 mammals, you still have some signal, but it's smaller. You don't have as much power at that level. But you could do something different. You could basically say, let me estimate the excess constraint, not just above my threshold, but over the entire stretch of that distribution above some, much lower threshold, okay? So the actual distribution of conservation scores, namely your signal, versus the expected distribution here in blue. So basically this is the, so how do you build this? You basically can build this by asking what is the distribution of evolutionary constraint that I observe empirically in regions that I know are not functional. So I could look at very, very short introns, or I could look at ancestral repeats that match the consensus. So all these regions allow me to now build some kind of distribution for what I would expect by chance, and that's the blue distribution. Then I look at the genome that I care about, so say the remainder of the genome, and I build the observed candidate element distribution, so that's your purple. And then you ask, what is the deviation of that purple distribution from the blue distribution? And you could basically say, what is my false discovery rate at any one position? Because I know that anything below the threshold that I choose is going to be called as non-conserved. Below that threshold is going to be called as non-conserved, and above that threshold is going to be called as conserved. So ultimately, every single time I do a classification, I choose some threshold, and then anything above that threshold, I say yes, anything below that threshold, I say no. Where I put the threshold changes my false discovery rate. And here I can tune my false discovery rate by basically saying, what fraction of the blue distribution am I willing to call as conserved? And that's my FDR. Is everybody with me here? So I can choose a very stringent FDR or a very loose FDR. And then I can basically say how much of the purple distribution is above that threshold. The problem, of course, is that the more stringent I become, the more of this gray bar I'm going to be throwing away. But if I instead look at the overall shape of these distributions, I can estimate that there's an excess constraint that corresponds to all of these different black regions, whether or not I have the power to detect them. Because in order to detect this one, I would have to have 80% false discovery rate, okay? Because, most of these regions under here are in fact, or many of these regions are coming from the background distribution. Raise your hands if you're with me. Awesome. Any questions? Yes?
So what do you mean by constraint, evolutionary constraint?
It's exactly what we've been looking at here. So basically, the fact that I don't observe any substitutions here, that basically tells me that evolution has been working to exclude the mutations there. Does that answer your question? Any other questions? Yeah. Yeah, so when it's the same thing as the reference up there, I put a dot. That means that it's conserved. When I put a different letter, it means that it's changed. Any other questions? Yeah.
When you're building the signal for the distribution for the signal, you build it at one nucleotide, is it for the whole?
So it depends. I can build it at the 50 nucleotide level, and then I have some distribution. I can build it at the 12 nucleotide level, and I can also build it at the single nucleotide level. Does that answer your question? Yeah, over the whole genome. No, over the whole genome. So basically, I take all of my ancestral repeats, and I build a distribution of 12 mer average conservation. Any other questions? Okay. So at any cutoff, there's a certain number of true positives, there's a certain number of false predictions, and we can't detect all of the constrained elements since the curves overlap. But we can estimate the total amount of excess constraints by integrating over the entire area between the two curves, okay? And if I use this approach on those curves here, I can basically say, well, here's my whole genome distribution, here's my ancestral repeat distribution, and here's my excess constraint. And for example, here you can see this rate, omega, for example. And if I look at the difference between them, what I basically see is that in the ancestral repeats, I have a more symmetric distribution. Whereas in the whole genome constraint, I have a longer tail on the, less mutations side. So this is purifying selection. This is selection against change, okay? And then by looking at the difference between the whole genome and the ancestral repeats, by subtracting the two distributions, I can basically say, here's my overall excess constraint. And at any one threshold, it's very difficult to detect that, but I can estimate the total amount of evolutionary constraint. Everybody with me? Awesome. So basically, you can measure both excess purifying selection, and also what we notice is that the whole genome curve has a longer tail as well on the positive side, on the more mutation side. And this is possibly positive selection. This is basically regions that have accumulated more regions and have selected these more regions, and that's probably adaptation to new environments. Okay. So also many new elements are in intronic and in intergenic regions. Okay, and the highest enrichment, however, is in coding regions. So yes, most of the evolutionary constraint sits outside protein coding exons because protein coding exons only cover 1.5% of the genome, but the highest enrichment is indeed found in those protein coding exons. Sounds good? So we can both ask, where do most of the regions fall, and also what are the regions that are most enriched for conservation. The other thing we notice is that even if you just ask, when I build my alignments, is there even an alignment in each of the species? The fact that there is an alignment already tells me that that region has not been lost. And many non-functional regions are likely to be lost. So just even having an alignment is actually important information. So basically, if you look at the depth of coverage in protein coding exons in blue, or in 4 mammals or 29 mammals, you basically see this distribution of coverage. Whereas if you specifically look at the distribution of coverage over ancestral repeats, you basically see that they're much less covered. Many fewer species even align in these places. And when they do align, they do have more substitutions. Everybody with me here? Awesome. So you can basically ask, what is the fraction of the genome that I can estimate to be under constraint, the fraction of the genome that is detectable at some 5% FTR, and the fraction of nucleotides that is detectable at some FTR. And what you can see is that as I increase the number of species from 4 species to 29 species, the fraction that is detectable at the 12 mer level and also at the 50 mer level is dramatically increasing. So here, nothing is detectable at the 12 mer level with only 4 species, but with 29 species, 5% of the genome is detectable. Oh, and we can estimate that there's in fact a total of 7% that is conserved. Sounds good? So, yeah.
You did, like, you first showed the yeast genome that are together, but you were comparing only 4 types of yeast, right?
Yeah.
So why do we see like 0% of this happening?
Where here?
Yes.
It's the question is across the entire genome, what does the distribution look like? And in any one location, yes, you can see this excess constraint, but across the genome, you're basically testing a lot of hypotheses. So for genome-wide discovery, you have no power. But in the case of the yeast, as I'm going to show throughout the lecture, we were using specific types of signatures that give you a lot more power. Okay? Sounds good? So now that we've talked about constraint at the nucleotide level and measuring regions that are under constraint, let's now discuss the patterns of constraint, so increasing the complexity here. So as I mentioned earlier, there's this funny region here in the middle, okay? Between exons 5 and 6, there's this region that shows very, very strong conservation, equal to all of the exons, but much less deep conservation, more like these intronic regions. So what is going on here? The question is, can we not only find what regions appear to be important, but also pinpoint the specific function within these regions? And then the answer is yes. We can not only recognize the amount of constraint, but also the pattern of constraint. Namely, the pattern of change that we see, the patterns of mutations, insertions, deletions, can in fact distinguish different types of functional elements, because the specific function that the element performs is what's being selected. And if that function is preserved, it doesn't matter whether the underlying nucleotides are changed. So the function dictates the selective pressure, which in turn dictates the patterns of mutation, insertion, and deletion. So the concept here is the following. If you have a set of identically functional sequences that all could fulfill the underlying function of, say, this protein. So this protein performs some function. And then I can explore different sequence encodings of that same amino acid sequence, but I can also explore different amino acid sequences that all have the same fold. This is basically exploring evolutionary, in evolutionary time, exploring the space of functional sequences that all map to the same function. And with more species, I have more points that I can sort of build the shape of that cloud that all maps the same function. And for protein coding regions, that cloud has a different shape than for RNA regions, and then for motifs, and so on and so forth. So basically, the shape of that space that evolution can observe, can explore, while maintaining the function, is in fact what allows me to infer these evolutionary signatures. So the fact that for an RNA sequence I can tolerate different types of mutations, that simply has to do with the shape of that space that all encodes the same function. Okay? Raise your hands if you're with me on that analogy. Awesome, great. So now we can develop evolutionary signatures that are characteristic of different types of functions. Okay? So what we're going to do throughout this lecture, and what we've done over the last 10, 15 years, is develop a series of evolutionary signatures for different classes of elements. For protein coding genes, these are codon-substitution frequencies and reading frame conservation. For RNA structures, they're compensatory changes that change here and there while maintaining the folding, or silent GU substitutions that maintain, for example, if you have an AU, you can change it to a GU, or if you have a GC, you can change it to a GU, because RNA pairing is more promiscuous than DNA-based pairing. For microRNAs, the shape of the conservation profile, structural features such as the loops and the pairs, or the relationship of these sequences with 3' UTR motifs, are all different types of evolutionary signatures that allow us to detect those. And then for regulatory motifs, the fact that the mutations preserve the consensus binding site of the transcription factor. The fact that these motifs that are conserved show an increased branch length score compared to non-functional k-mers. And the fact that they're conserved not just in one location, but in hundreds of locations across the genome, together give us signatures that can allow us to recognize these regulatory motifs. Okay? So overall, we can in fact ask which of these signatures is applying in the region of evolution of alignment that we're looking at, and therefore what selective regimes does that region appear to be after, what is the space of sequences that all encode the same function, and from that infer the distinct classes of functions. Is everybody with me here? Raise your hands if you're following the concept of evolutionary signatures. Awesome. Any questions? Okay. So we've basically used that, again, over the last 10, 12 years to basically discover new protein coding genes, to revise the gene annotations of the yeast, the fly, the human, and the worm genome, to detect unusual gene structures that defy the rules that we knew about from the central dogma before, to discover new structural families of RNAs, to recognize targeting, editing, and stability motifs, to discover ribose switches in mammals, to expand the existing microRNA families in fly and in human, and to discover new microRNA families, to discover new properties and new types of function of microRNAs. For example, when the two arms of a microRNA can cooperate, or when the same region of DNA can be transcribed in two opposite directions that both encode different microRNAs and providing these switches, to discovering new regulatory motifs, to identify individual regulatory motif instances, to build networks of transcription factor regulation and microRNA regulation, and to also recognize motifs at single binding site resolution. So the power of these approaches is truly immense. Okay? So now let's dive into the nitty-gritty details. How do we actually build these evolutionary signatures? So we're going to start with protein coding genes, and then I'm going to go briefly over RNAs, microRNAs, and motifs. Okay? So in this region lies exon-intron boundary. Half of this region is protein coding. The other half is non-coding. And my question to you is, which half is protein coding? Okay, raise your hands if you can see the answer. Raise your hands if you cannot see the answer. Awesome, this is great. So stare at this region. What do you find? What do you see? What's funny? There's some funny things that distinguish right from left. I have one answer, two answers, three answers, four, five. Okay, so go ahead. Raise your hands if you could see that before. Awesome, great. So on one side, I have a gap of 3, and a gap of 6, and a gap of 6. They're all multiples of 3. That's kind of cool. On the other side, I have a gap of 1 and a gap of 5. They're not multiples of 3. Okay? Good. So one difference between them is the length of the gaps. If the gaps are modulo 3, 0 modulo 3, then we're good. If they're multiples of 3, we're good. Okay? What else do you notice? From the hands that were up before, everybody wanted to point out the frame preservation? Yeah.
At the beginning of the sequence is very atypical.
Yeah. Very cool. So that's a sequence property. It's a property of the primary sequence, and that's kind of cool that you noticed that. But I'm looking for evolutionary properties, something about the patterns of change. But you're absolutely right. And non-coding regions tend to be a little more AT-rich, and protein coding regions tend to be a little more GC-rich. Count the number of stars. How many stars in a row do you have? On the left and on the right. Anybody notice anything funny about the stars? Yeah.
The right side is patterns of 2 stars, which is similar to like the generacy of amino acid coding.
Very cool. So if you look here, you basically have conserve, conserve, not conserve, 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 not conserve. What I like to call bang, bang, poof, bang, bang, poof, bang, bang, poof. Yeah, you see bang, bang, poof, bang, bang, poof, bang, bang, poof, bang, bang, poof. Okay. So that basically tells you that there's some kind of periodicity. There's a 3 periodicity. The first 2 positions, presumably, are more conserved, and the third position, again, presumably, is less conserved. Who can see this in retrospect? Okay, very cool, awesome. So I'm now going to draw a vertical line and basically tell you that indeed, to the right side of that line, the gaps are multiples of 3, whereas to the left side, they're not multiples of 3. And then, if I now draw the boundaries of the codons, you see these incredibly strong signals, bang, bang, poofness, of conserve, conserve, not conserve, right? So what's happening here? Evolution has 2 components. Mutation is really blind. It's just throwing mutations everywhere. But selection is excluding those mutations that would disrupt the function of that region. And the function in this particular case happens to be at the protein coding level, okay? So the gaps are multiples of 3. The mutations are largely 3 periodic. And specific triplets are exchanged more frequently. Even when you have these substitutions are happening to amino acids that preserve the function of that region. And again, you have here a splice site, which is switching between this non-coding and this coding region. Now that you see the coloring, who thinks that this signal is overwhelmingly strong? Raise your hands. Right? Awesome. So basically, that basically says that, yes, these signatures are there. You just need to know what to look for, okay? So these are the different signatures of protein coding selection. And as we start quantifying these, you're going to see how much more powerful they're in fact getting. So, testing. Let's see, testing, the sound is working, yeah. Okay, so the first signature of these gaps being multiples of 3 has to do with reading frame, obviously. So if I write the fat cat sat, you understand the sentence. If I delete 1 letter, the at makes no sense. If I delete 2 letters, the makes no sense. And then if I delete 3, the cat sat still makes sense. I've lost some aspect of that sentence. Now, it might not be about Garfield, but it still makes sense, okay? And in the same way, when I'm translating amino acids, losing 1 amino acid is fine, but mistranslating all of the remaining amino acids is really terrible. So DNA insertions and deletions that preserve the amino acid frame are much more tolerated, okay? And then on the bang bang poofness side, the patterns of mutation are such that the genetic code organization is preserving the amino acid translation. Namely, the way that amino acid translation is organized, the way that the genetic code is organized, it is very common to have 4-fold degeneracy, where the first 2 nucleotide positions of a codon are sufficient to, in fact, fully determine the codon. And the third codon position to fully determine the amino acid translation of that codon. And the third codon position is not as important, okay? And that has to do partly with the tRNA usage, where the genome is kind of being cheap, and instead of encoding all possible codon, tRNAs for all possible codons, sometimes it'll say, well, I just need to pay attention to the first 2 letters, and then the third one will be a wobble position, which will kind of make loose contacts and recognize any of the 4 nucleotides. So part of that is that the organization is such, part of it is that the tRNAs themselves are sort of, you know, being kind of cheap. And the end result is that mutations that preserve the first 2 positions but disrupt the third are perfectly tolerated by evolution in a protein coding group. Raise your hands if you're 100% with me here. Awesome, great. So now let's scan these regions for different types of change, okay? The previous one I had done by hand. This is now done by the programs that we have developed in my group for scanning for these protein coding regions, okay? So here is the nucleotide level conservation of this region. And you can see, yeah, there's some stronger constraint within protein coding exons, but there's a lot of other constraint outside protein coding exons, right? And even protein coding exons sometimes have less nucleotide level constraint. So if you only looked at these track, it would be very difficult to pick out exactly where the protein coding regions are. Everybody with me? But instead, what I can do is actually start coloring synonymous substitutions. These are substitutions where you change the codon, but you preserve the amino acid translation. Or conservative substitutions, where you change the amino acid, but you preserve the properties of the amino acid. Or non-conservative, where you've actually disrupted the properties. I can color frame shifts, and I can color the 3 different subcodons, okay? And if I do that, this is what these 2 regions look like. So this region here, which is, you know, kind of well conserved, and that region here, which is kind of well conserved, are basically showing completely different signatures when I put in the lens of evolutionary signatures. So in this particular region, I have a lot of synonymous substitutions. In this particular region, I have a lot of non-synonymous, a lot of frame shifts, stop codons appearing, and so on and so forth. Yeah.
Well, I understand how you can look at the frame shifted regions there. Are there like indels that just been removed?
Yeah. Basically, right now, this is, you don't see the gaps, but they are the ones causing frame shifts. Thank you. And then I can apply that systematically across a larger region. And this is what you see, okay? Remember when I asked you earlier, who can find the protein coding gene? Who can find it now? Raise your hand. Awesome, right? It doesn't take a statistician, just like it's there, right? It's magnificent. Basically, the genome comes to life when you shine the light of evolution on it. So we're asking evolution, hey, what does your notebook say about all these experiments you've been performing? And evolution is like, well, you know, when I mutated this nucleotide, it was perfectly fine. And when I mutated that nucleotide, it was not okay, and so on and so forth. Everybody with me? So now you can basically discover protein coding genes with amazing sensitivity and specificity, okay? So how do we implement it? For reading frame conservation, we can basically simply ask, well, what fraction is in the same reading frame? So if I have an indel here, then that region has at most 55% reading frame conservation. And that region here, 90%, and that region here at most 40%, and that region here at most 60%, and so on and so forth, because I've sort of shifted the frame, okay? And I can use that to basically ask, well, how can I, basically what is the best possible alignment between frame alignment, between the reference sequence and my alternate sequence, where if this switches between frame 1, frame 2, frame 1, frame 2, frame 3, and then at most I have 43%, 34%, or 23% in the same frame, and then I can basically score that across all of these different regions and basically get an average of 56% conservation, which is not much different from random, versus 100% conservation from regions. Everybody with me here? So using only this reading frame conservation test, we applied it to yeast, and we found that in 4,000 genes that had a gene name, we could actually keep 99.9% of them. And for 300 randomly chosen intergenic regions, we could reject 99% of them. This is pretty awesome in terms of sensitivity and specificity. And then we basically applied this test to 2,000 hypothetical open reading frames, which were long enough to look like a gene, but didn't quite yet have a biological function yet. And what we found is that 500 of them were very strongly rejected. Here's one example. You basically see all of these indels that are not preserving the frame. You see all of these non-conservative substitutions. You see that there's no pattern of bang-bang poofness, where basically here you see this periodicity of green, green, yellow, green, green, yellow, green, green, yellow, green, green, yellow. And then in non-coded regions, you just don't see that at all. And this is something that people thought was a protein coding gene before. They were doing hundreds of experiments on it to figure out what it does, and no one had figured out what it does. And the reason is that it's just simply not a gene. It was just a non-coding region that just simply happened to have no stop codons. Okay? So that's just the reading frame score. But we can also utilize these nucleotide substitution patterns that I showed you on the previous slide. Okay. So what would we like in such a method? We'd like to quantify the distinctiveness of all 64 square possible codon substitutions. Synonymous should be very frequent in protein coding sequences, and nonsense should be much more frequent in non-coding than in coding regions. We want to model the phylogenetic relationship between the species, the actual tree that relates to species, where multiple apparent substitutions could be explained by just one evolutionary event. We'd like to tolerate uncertainty in the input because we don't know the ancestral sequences. We'd like to tolerate gaps and missing data. And we'd like to also report both the confidence in our result and how uncertain we are. Quantify the confidence that the given alignment is indeed protein coding in some unit of either a p-value or bits of information and so on and so forth. So basically what we did with Michael N is to basically model codon evolution as a Bayesian network, where you basically have some ancestral unknown internal nodes and some observed extant species that we can sequence. And on that phylogenetic network lies an evolutionary matrix that basically tells you how, with what frequency am I replacing each codon with each other codon, each triplet of nucleotides with each other triplet of nucleotides. Raise your hands if you're with me so far. Awesome. So basically every site, every codon alignment column is treated independently, and given the topology of the tree and this conditional probability distribution, we can simulate evolution of an ancestral sequence, and given some, so that's the generative part of the model. Basically, I can run the model forward and basically see what types of mutations do I obtain. Or given observed leaf sequences, I can infer the most likely state of the ancestral sequences, the maximum posteriori probability sequence. And there are, you know, a lot of parameters. Basically, if I have L leaves, I basically would have 64 square parameters for every leaf, and for every branch, and then 2L minus 2 internal branches. So that's a huge number of parameters. Instead, what I could do is simply say that there's a single substitution matrix that I want to estimate across the entire genome, and then apply that at every branch over and over and over again. So we can basically parameterize this conditional probability distribution using a rate matrix, which is now shared throughout the whole tree, okay? And at every branch, that rate matrix is scaled to the power of the number of events that have occurred, the amount of time that has elapsed. And at every event, I just multiply this matrix by itself. Is everybody with me here? So basically, if I have 1 unit of time, I multiply this matrix alone. If I have 2 units of time, I multiply this matrix on top of that matrix. And that basically gives me 2 substitution events. Raise your hands if you're with me. Awesome. So basically what we want is the probability of having the child, it's a generative model, given the parent and given the time that has elapsed, which is some scaling of that matrix to the teeth power, okay? So the intuition is that the branch lines specify how much time has passed between any 2 nodes, and the rate matrix describes the relative frequencies of codon substitutions per unit branch time. And then the synonymous substitutions have high rates, and the nonsense substitutions have very low rates, okay? So here's one example of a 4x4 matrix and how you can run it, you can scale it, and you can basically look at the frequency of substitutions as you go through, okay? So the challenge, of course, is to estimate Q and figure out a Q that can be exponentiated to match this unit time matrix, okay? And, you know, there are strategies for doing that. But after you've done that, you've basically obtained your rate matrix Q, and you can now train one such rate matrix for different classes of elements in the genome. So if I apply the same rate matrix to, you know, coding and non-coding sequence, I can basically ask how likely is it to be generated. But much more interestingly, I can estimate a coding matrix QC and non-coding matrix QN. And then I can say which of the two models of the world better fits the data. Okay, so I'm going to basically do a log odds ratio of how much more likely is it that the data was generated according to the coding matrix or to the non-coding matrix. Raise your hands if you're with me. Awesome, great. So now I can basically say, what is the probability of observing this sequence given the coding matrix? What is the probability of observing that sequence given the coding matrix? And conversely, what is the probability of observing that sequence given non-known coding matrix and given that sequence given the non-coding matrix? Okay, so notice the difference in these exponents. So basically this is 10 to the 30 times more likely according to a coding model. 10 to the 30. I mean, that's superbly strong. And this is 10 to the 20 times more likely according to the non-coding sequence. Everybody with me here? So of course these numbers are very small because there's a huge number of possible alignments that my generative model can generate, but it's the likelihood ratio that you care about, okay? So again, 10 to the 35 and 10 to the 21 is the difference in ratio. Okay, so that basically means that this alignment is 10 to the 35. That's 10 followed by 35 zeros. I mean, I don't know if you get the magnitude of that. I mean, my 4-year-old daughter last year was basically saying, is 100 even a number? Because she couldn't count to 100. But I would ask, is 10 to the 35 even a number? It's just so big. So anyway, I'm in awe in front of huge likelihood ratios. And that basically means how powerful the model is, how much evidence there is. I mean, I don't know if you guys remember, but 10 minutes ago, you were staring at this sequence and you were like, can't tell what's coding and what's non-coding. And this is 10 to the 35 times more likely to be coding once you have the right probabilistic model. Okay? Who thinks this is kind of cool? Yay, awesome. So now, let's just go crazy with it. Let's apply it to do all kinds of really good in the world, okay? Let's revise genes, let's discover new mechanisms of gene function, and so on and so forth, okay? Remember how I told you that recognizing protein coding regions using just nucleotide conservation was really no good. You know, there's this region here which is very highly conserved, there's this region here which is clearly known to be protein coding but not very well conserved, and so on and so forth, okay? Let's now run our protein coding evolutionary signature. I mean, this is striking. You can see how clean this signal is and how clearly it picks out protein coding regions. So this is just basically applying exactly what I showed you on the previous slide. And you can then apply some kind of hidden Markov model to decode that sequence into a set of exons that are predicted to be protein coding. When you do that, you basically see that you can perfectly capture all of the previously known exons and discover a new exon, which is, you know, not previously annotated, right? And indeed, when you do the right experiment and you can sort of fish out transcripts that have that region, you find that indeed there's an alternative transcript of that gene that indeed captures this particular exon rather than that particular exon, okay? So we use that to basically capture many new genes and reject previously annotated genes, okay? Here's one example of a new protein coding gene in the human genome. You can see here there's an ATG, there's this beautiful protein coding constraint, there's this stop codon conservation, and then surrounding it, you have frame shifts, you have stop codons, you have all kinds of crazy stuff, right? Super powerful. What else can we do? We can basically look for places that disobey the rules that we've learned so far, right? Like all genes start with a start codon and stop with a stop codon, right? This is like the most basic rule. Well, here's a stop codon. Does protein coding constraint seem to stop here? No. Here's another stop codon. Does it seem to stop here? Yes. So this is kind of like driving in Athens, right? You basically see a stop sign. If there's no cops, just go straight through, okay? This is like the Greek driver. So what's going on? We basically found that there's about 300 genes in the fly genome that appear to be ignoring their stop codons and be perfectly well conserved thereafter. These are deeply conserved stop codons, and yet they're simply ignored. There's this joke in Greece where basically, you know, this guy with a huge truck is driving through and he just runs right past the stop codon. The policeman says, "Hey, didn't you see the stop codon?" And the guy replies, I saw the stop codon. I didn't see you, Mr. Policeman." So yeah, in this particular case, yeah, well, they appear to not notice the policeman or not notice the stop codon. So what we found is that these were very strongly enriched for neurotransmitters, for brain-expressed proteins, for transcription regulators. They were, you know, they had specific upstream bases that show increased conservation. Downstream was much more likely to be a TGAC. They had GCA triplet repeats. They had increased secondary structures. So there seems to be a controlled, regulated mechanism through which these hundreds of fly genes are ignoring their stop codons. And we also found several candidates in mammals that sort of appear to have this stop codon read-through. So we basically found also experimental evidence of protein translation downstream. And again, this is a completely open field. This is something that was never recognized to be such a prevalent phenomenon before we were able to sort of detect them systematically. Yeah.
So we have this idea of every codon translates to amino acid. What do we know what happens at a
Such a great question.
level?
So there's something called selenocysteine, like the 21st amino acid, where you know, this is sometimes incorporated, but these are not selenocysteines. You can actually pull down all selenocysteines in the genome, and these are not selenocysteines. So something else gets incorporated there. The current consensus is that it's just a random amino acid. We don't really know which one gets incorporated. And
Do we know if something gets incorporated, or
yeah, you could also keep reading. So these events are infrequent enough that we don't have spectra that specifically span that, but this is still a big question. So it's a great question. Other questions? All right. Here's the other thing that you can do. When you start modeling evolution so exquisitely, you can basically say, well, what else is not being obeyed? And in some cases, we find that in protein coding regions, you like to see this. You like to see a lot of green. Green basically tells you that mutations are happening, and those mutations that are happening are preserving the amino acid properties, okay? But there are other regions where there's no green. Yes, there's no red, but there's no green either. So there's no amino acid disrupting mutations, but there's also no mutations period. And that allows you to now start asking about the synonymous constraint, not just the amino acid constraint, but also the synonymous constraint. And as you scan through the genome, you can basically estimate not only the amino acid constraint, but also the synonymous constraint. And in some places, the synonymous rate drops down to 0. There's like no mutations there. And what we found is that out of all of the Hox proteins, these are the proteins that govern the anterior posterior developmental axis. They're arranged in the same order that they're expressed. They form 4 clusters in vertebrates, but only 1 such cluster in flies, and they're all in the same order and orientation. These genes appear to have a huge number of these synonymous constrained elements, where the synonymous rate in the middle of the protein drops to 0. What's happening there? Well, you see, there's in fact matches for a large number of regulatory motifs, which are known to be controlling the patterning of these genes. So again, there's overlapping constrained elements. And we were able to use this approach to find hundreds of these elements across the genome. You can basically now predict overlapping protein coding genes. You can find RNA structures encoded within protein coding genes and so on and so forth. And just a few weeks ago, we published this paper in Genome Research. It hasn't even appeared in print yet. It will be in the December issue. That basically said that here's a new catalog of protein coding genes in the human genome. We basically were able to discover hundreds of new exons using the same Phylo-CSF approach, phylogenetic codon substitution frequency approach. And in many cases, in about 118 cases, non-coding GWAS SNPs were falling right in the middle of our new protein coding exons. So, you know, we talked about how there's all these non-coding variants that remain to be annotated. In this particular case, here's a SNP that was previously unannotated. It's associated with myopia. And then there's a Phylo-CSF region that exactly overlaps it. Here's the GWAS Manhattan plot. And you can basically see that the strongest genetic score sits exactly where the new protein coding exon sits. And that is an alternatively spliced exon, which is only expressed in the eye, which is kind of cool, right? So, and it's ironic that it's associated with myopia because, again, you know, being able to sort of have better lenses for evolutionary interpretation might actually help us get better eyesight one day, which is kind of cool. And then you can see here that there's hundreds of these, like more than 100 of these across the genome associated with just a huge number of different traits. So this has major, major implications. Being able to actually discover where the protein coding genes are has major implications for understanding disease. Okay?
That's a question.
Yeah.
That's a really good one. Can you go back? So it says traffic over there, like in the first figure. Leukemia.
Trafficking.
Okay.
So not traffic, sorry. This is not about Greek driving.
I was like, okay.
So we focused a lot on protein coding signatures, but again, as I mentioned earlier, we can build these signatures for many different other kinds of elements. When we spoke about RNA folding, we spoke already about these RNA signatures. When we spoke about regulatory motifs, we spoke about these motif evolutionary signatures. So, you know, we can build these phylogenetic stochastic context-free grammars that basically generate these production rules with different probabilities. And these production rules basically produce pairs of columns of the alignment that allow you to infer these evolutionary signatures for non-coding RNAs. We were able to use that to discover many new RNA structures and many new RNA families. For motifs, I mentioned previously this motif conservation score, the fact that not just one instance of the motif is conserved, but hundreds of instances of the motif are conserved across the genome, and there's specific signatures, like whether they're conserved in intergenic regions versus coding regions versus upstream versus downstream. And these signatures allow us to now discover k-mers that are preferentially conserved. And then for transcription factor binding sites, we also in the motif lecture talked about how these show increased phylogenetic length and preservation of the consensus while tolerating movement, and how we could translate the overall amount of conservation into a confidence score by shuffling these motifs and getting an estimate for the expected amount of conservation for noise motifs versus the signal of these motifs. And then for microRNAs, again, the challenge is that there's only 100 true microRNAs, but there's 760,000 microRNA-like hairpins. So how do we distinguish the two? Again, evolutionary conservation can help us by basically building this profile of what is the conservation pattern of these hairpins. And what we find is like lower conservation surrounding the hairpins and lower conservation in the middle of the hairpins, but higher conservation within the body is in fact the signature for microRNAs. And that gives us one such signature, but then we can combine that with structural features, with other features together. And that gives us basically this 4,500-fold enrichment for recognizing these true microRNAs. So you can basically build trees for developing those and for recognizing those, and we found all kinds of really cool properties of microRNAs. For example, within the same Hox clusters that I was mentioning earlier, we found that there are antisense microRNAs, and also where the same region can be transcribed in two different directions, and then they control very, very different processes. And by changing the expression of one of those microRNAs, we were able to change a whole organ of these flies from these flight balancing organs to actually a wing, which is quite remarkable developmentally. And then this is thanks to this switch that is implemented by these sense-antisense microRNAs. The last thing is measuring selection within the human lineage. So mammalian constraint, in fact, matches human SNPs in some cases, where you can basically see that the SNPs that are happening in the human genome are in fact sometimes observing the mammalian constraint, which basically says that evolution is fine and well and still working within the human lineage. And using that, we can basically start asking what is the pattern of substitutions that we see in different classes of elements. And by estimating the overall constraint, even though we don't have the power to detect any one region within the human lineage, we can estimate the overall constraint in active regions. And then that allowed us to basically say that from the distribution that you would have expected in all these different classes of elements, if you had a motif that was bound, you basically had within the human lineage dramatically more constraint based on the proportion of derived alleles than what you would expect by chance. So that allowed us to now start estimating the amount of constraint in different classes of elements. And also within different motifs, you can basically see that if you add up all of the instances of the CTCF motif, for example, you basically see that the instances that are bound are in fact showing more constraints specifically for the high information content positions, whereas the sequences that are not bound are showing less constraint. And that we only get power because we have hundreds of these instances across the genome, so you can leverage all of that information jointly. So what we talked about today is nucleotide conservation and the concept of purifying selection, how it works, how to detect constrained elements at the nucleotide level, window level, and using HMMs, how to estimate the fraction that's constrained by looking at these distributions of signal versus background. We talked about evolutionary signatures beyond the primary sequence and beyond the overall amount of conservation. What is the pattern of evolution? And the more mutations we had, the more power we had. We focused a lot on protein coding genes. We looked at reading frame conservation and also codon-substitution frequencies as the two key signatures for protein coding evolution. We saw how you can get 10 to the 30 difference, even for small regions, between this QC model of the world and this QN, this coding and the non-coding matrices, and then how this concept of evolutionary signatures generalizes to many other domains to discover RNAs, regulatory motifs, individual binding sites, microRNAs, and sort of all the things that you can learn from that. Who feels that they've learned stuff today? Awesome, great. See you guys on Thursday to turn this loop around and now learn about evolution using comparative genomics.